So first we'll see about the formation of seed from ovule. So how the seeds are formed from ovule. So seed, what is seed? Seed is the fertilized ovule. So seed is the fertilized ovule that is formed inside the fruits. So we saw that after fertilization, so what are the changes? First, we saw the structure of ovule, right? So what are the structures that are present in the ovule? In the middle is the embryo sac, and that is surrounded by the nucellus and then outer integuments. So all these structures we already saw. So all these structures which are present in the ovule, now it is converted into that is ovule as such, it is converted into a seed, right? So seed is nothing but the fertilized ovule which is formed inside the fruit. So the ovary is converted into a fruit. So it is the final product of sexual reproduction, right? So it is the final product of sexual reproduction. That is, it is the product that is formed after fertilization. That is the fusion of male and female gamete, right? So these seeds, they consist of seed coat, then cotyledons and embryonal axis. That is the axis. So these are the structures of different seeds. So it is a dicotyledonous seed, that is 2 cotyledons, and they have the micropyle, right? So here the seed coat, outer is the seed coat, and then this is the endosperm, and then this is the cotyledon and hypocotyle. It is a root axis, and down there is a root tip, and which is covered by the root cap, right? So this is the shoot apical meristem which grows upwards. And here if you see, this is this. So these are the examples for some of the dicot and the monocot seeds. Okay, so this is the seed coat and cotyledon. This is a shoot apical meristem, then hypocotyle, and then root tip. So this is the endosperm. So these are the structures, that is all the seeds, they have the seed coat, cotyledons and the embryonal axis. Okay, so the cotyledons are simple, generally thick and swollen. So because they are rich in food reserve, right? Because due to the strong food as in legumes. So the cotyledons are simple, generally thick and swollen due to the food reserve, that is due to the presence of the food, right? So this is the case of monocot, that is they have the scutellum and then coleoptile and coleorhiza. So these structures we saw yesterday, right, about the structure of the embryo, that is monocot and dicot embryo. So if you take the seeds, there are 2 types of seeds. One is a non-albuminous seed and another one is the albuminous seed, right? So the non-albuminous seeds, which are also called as non-endospermic seeds, which are also called non-endospermic seeds. Non-endospermic, which is also called X-albuminous. X-albuminous seeds. So these seeds, non-albuminous seeds or non-endospermic or X-albuminous seeds, they have no residual endosperm. So endosperm, we saw how it is formed after fertilization, right? So there will be no residual endosperm as it is completely consumed during embryo development. So what is happening? This embryo, right, after the zygote, so they have the no residual endosperm. So there are 2 types of seeds. One is a non-albuminous seed and another one is an albuminous seed. And in the case of non-albuminous seeds, they have no residual endosperm as it is completely consumed during the embryonic development. And in the case of albuminous seeds, they retain some endosperm. That is, even after the utilization for the embryo development, the seeds will have some endosperm. So examples for the non-albuminous seeds are pea, groundnut. Sorry guys, sorry for the inconvenience. So there are some internet issues. Okay, we'll continue. So today you're going to see about the post-fertilization events, that is about the structure of the seed and about the structure of the fruit, and then about the apomixis and polyembryony, right? So under seeds, we saw that seed is a fertilized ovule which is formed inside the fruit, and it is the final product of sexual reproduction, and it consists of seed coat, that is cotyledon and embryonal axis. So the cotyledons are actually very simple, and they are generally thick and swollen, and due to the presence of the food reserves, right? So, and based on the endosperm, that is the mature seeds, whether they contain the endosperm or not, they are classified into different types of seeds such as non-albuminous seeds and albuminous seeds. So the non-albuminous seeds are also called as non-endospermic seeds or egg albuminous seeds, egg-albuminous seeds, right? So the examples here are pea, groundnut, and beans. So these are the examples for the non-albuminous seeds. So why are they called non-albuminous seeds? Because they don't have the endosperm. So they are also called non-endospermic, because all the endosperms will be utilized during the embryo development, right? So, and then the next category is the albuminous seeds. So what are albuminous seeds? So from the name itself we can know, so there exists some endosperm inside. So they retain some endosperm, which are also called as endospermic seeds, right? Which are also called endospermic seeds. So examples for these albuminous or endospermic seeds are wheat, maize, barley, castor, and sunflower. So these are all examples for the albuminous seeds or endospermic seeds, right? And also we have already seen about the coconut. So coconut, the liquid that is present is the nuclear endosperm, and the surrounding white kernel is the cellular endosperm. So yesterday we saw, right, about the endosperm, and also about the different types of endosperm, such as the nuclear endosperm and cellular endosperm and halobial endosperm, right? So, and about the types of the seeds.